Cool. All right, well, let's get started. So today, again, we're just continuing our lecture series in neuropharmacology. Yesterday we talked about depression, and today we're going to talk more about anxiolytics and hypnotics. So these are drugs that, again, like it says, anxiolytics, usually used to kind of treat anxiety-like symptoms, but a lot of these drugs as well that we're talking about also have a hypnotic effect. So what do I want you to get out of this lecture? So similar to the depression one yesterday, again, I know it's not a neuroscience unit, but I still think it's very important, at least just to have some minimal understanding of what kind of changes you experience or see in the brain in people that are experiencing anxiety disorders or anxiety-like symptoms. Very, very minimal amount of neuroscience there, so don't be turned off too much by it. And then we'll move into the pharmacology, especially about the treatments. We're going to talk about mostly benzodiazepines because they're the most common prescribed anti-anxiety drug. Then we'll briefly touch on etifoxine, which is another different type of anti-anxiety drug. And then we'll move on to focusing more on hypnotics. Mostly we'll focus on barbiturates, and then we'll end the lecture by looking at melatonin receptor agonists as well, which might be relevant to a lot of people that take things like melatonin to help them fall asleep. Similar, I don't have the disclaimer here that I had yesterday, but similarly, everything I'm talking about here is just for educational purposes. It's not for clinical diagnosis or anything like that, so just bear that in mind. So again, this is on anxiolytics. So mostly anxiolytics are used to treat anxiety disorders. So what are anxiety disorders? Well, anxiety, is a large group of mood disorders. It can be very, very complex, similar to depression. But anxiety disorders can, for example, characterize any of these different disorders as well. So you have generalized anxiety disorder, which is probably the most common form of anxiety disorders. So this is where you have generalized anxiety just in general, so an increased physiological response, like a stress response. I won't get into kind of all the different symptoms. We're just going to mostly just lump them all together. But it can also involve, things like panic disorder, social anxiety, obsessive compulsive disorder or OCT, also trauma and stressor-related disorders, so post-traumatic stress disorder, which is really common in people that have experienced traumatic events in their life. And it also includes phobias as well, so things like arachnophobia, agoraphobia. So it's anything that really causes an anxious-like response in your body, so like a physiological response that kind of resembles stress or fear in situations that don't really call for those symptoms. So this can affect your daily lives in the form of things like panic attacks, physiological stress responses again, so you start sweating, your heart rate starts increasing, you might feel dizzy, faint, and then also this kind of has a psychological component as well, because obviously your body doesn't want to be in this kind of stress-like state, so you tend to kind of avoid situations that might generate those anxious feelings. And this can be really disruptive to daily life, so things like phobias like agoraphobia or arachnophobia, you might avoid places that kind of trigger that response. So it's a very intrusive mood disorder, and it kind of also has very high comorbidity with other mental health disorders, especially depression. Depression and anxiety can sometimes go hand in hand in a lot of people. So people that experience depressive symptoms also may experience anxiety-like symptoms. And it's a very high prevalence in the just in society as well. So one of the papers I was reading a while ago was saying that anxiety and depression together make up kind of like the most common form of mental health disorders, and it can be experienced in up to 30% of individuals. So it's a really high kind of economic burden. And just like I showed you this paper before about how depression prevalence has seemed to increase since COVID-19, anxiety disorders have also increased since COVID-19 as well, so increase in anxiety symptoms, which needs to be tackled. So when we're talking about anxiety disorders, what's really happening in the brain that's triggering this? And just like depression, there's no easy answer. So we see a lot of different changes in the brain, similar to depression, but there's not one underlying mechanism that's causing it. There seems to be a lot of different systems that are being affected. So neuroendocrine, so like kind of hormone systems, neurotransmitters, so things like serotonin, monoamines, glutamate, GABA is also changed. The connection between brain areas might also be different, so the communication is a little bit altered. And we think that these also contribute to anxiety disorders similar to depression. But most of the kind of changes that we see in anxiety disorders are thought to arise from disruptions in specific areas of the brain that are important for emotional processing. And this is just one kind of brain map showing you some different parts of the brain that might be affected. Again, we're generalizing many different anxiety disorders at the moment, but these are some of the key areas that are generally seen to be changed in some way. So the prefrontal cortex, again, similar to depression. So prefrontal cortex is around the front of your brain. Very important for executive functions, so things like motivation, I'm blanking on the word right now. Motivation, kind of controlling all your emotions, figuring out what is the correct response to do in certain situations. That's all controlled by the prefrontal cortex. The anterior cingulate cortex pops up a lot. It's kind of somewhere around here, just a little bit below the prefrontal cortex. Again, really important for emotional processing, learning, and kind of error detection as well. But you see this brain region pop up a lot in a lot of anxiety disorders. And then you also see a lot of changes in the limbic system. So I don't know if you've heard of the limbic system before. This is kind of just like a blanket term that we give to a lot of brain regions. They're usually a bit subcortical, so under the cortex. And there's a few different brain regions involved, but they're all really important for things like emotional processing and memory. And two kind of brain regions that always pop up a lot is the amygdala and hippocampus. So I don't know if you've heard of the amygdala before. This is the part of the brain that's really important for emotional regulation, mostly the fear response. So you usually hear, like especially in rodent studies, you'll see a lot of like kind of fear models and fear conditioning models also affects the amygdala, and we see similarly that in humans as well. And then also the hippocampus. So the hippocampus is really important for learning and memory. And the hippocampus has a lot of projections and talks a lot to the amygdala. So that kind of, if you have an altered fear response in the amygdala, that can start inducing a lot of maladaptive fear memories. And then if your prefrontal cortex or anterior cingulate cortex is also not working, you can start seeing some really kind of alterations in that mood and kind of appropriation of a specific fear response. And this is just showing you kind of a map. So this is functional MRI. I talked about it earlier. Basically, you get put in an MRI machine, and basically it looks at the blood signal. And basically, if you see an increase in a blood signal in one area, do you see another increase in the blood signal in another area? And that's just a way of saying these two regions of the brain are talking together at the same time. And so in a person with PTSD, which is this individual here, these are all the brain regions that are kind of active at the same time. And these are different to kind of people that don't experience PTSD. So you can see a lot of those brain regions that I talked about. So the amygdala is implicated on both sides of the brain. Anterior cingulate cortex. There's a few other regions as well, which we won't talk about. But then if you look at someone that also has another anxiety disorder, this one's social phobia, you can see a lot of the same players are affected as well. So you've got the thalamus is affected, it's talking to different brain regions, not as much as PTSD. So there seems to be, again, I don't want to hierarchicalize and say which anxiety disorder is worse than the others. I'm just going off this specific individuals here. But it seems that in this person with PTSD, there seems to be a lot more altered connections compared to someone with social phobias. But I'm just trying to illustrate that a lot of these brain regions are kind of linked together, so there's alterations in them, and you can, again, just showing you the complexity of anxiety disorders. Some have more altered connectivity compared to another. And just briefly, so what else is kind of altered in people with anxiety disorders? What do we think kind of happens? Well, we know that the hypothalamic pituitary axis is a little bit altered in individuals with anxiety disorders. So this is a neuroendocrine negative feedback loop that kind of regulates the levels of stress, hormones in the body and kind of your body's response. And this is controlled by glucocorticoids and mineral corticoids. So you've probably heard of cortisol before. That's a glucocorticoid. So what happens in the body is that usually when you have an increased stress response, your hypothalamus releases corticotropin releasing hormone. This causes the anterior pituitary gland to release adrenocorticotropic hormone. And then this works on the adrenal cortex, which is located more in your torso, to release these glucocorticoids. And you have this feedback loop. So when you get an increase in glucocorticoids, this inhibits the anterior pituitary from producing more of that stress hormone. And it also inhibits the hypothalamus from kind of producing more of that hormone. So your body has its own system in place. So when you release or have a stress response and release these stress hormones, the body realizes, okay, we have an increase in stress hormones, it causes that stress response, but then it tells your brain, okay, stop releasing this hormone after the stress is not encountered anymore. So you can regulate that stress. Because we all feel stress and, fright and fearful situations every day in our lives. But we're able, well, if you don't have an anxiety disorder, you're able to kind of resolve those feelings. But in people with anxiety disorders, these get affected. This feedback system is affected so that you kind of have an increased global stress response all the time. These glucocorticoids are important for a range of cellular processes. Again, that stress response, they play a role in inflammation and also cell metabolism. And these receptors are highly expressed in a lot of different brain areas. So this is just a map showing you where some of these receptors are located for these stress hormones. And as you can see, there's a lot of, this is in a mouse or rat brain as well. As you can see, there's high expression of these receptors in the prefrontal cortex, other parts of the frontal cortex, amygdala around here somewhere, and then hippocampus as well up here. So a lot of those brain regions that we see are altered have high expression of these stress receptors. So maybe this is how it's affecting the brain and causing plasticity and changes. These stress hormones can modulate neurotransmitter release as well, and it's just important to note that there's differences in the stress response between males and females and how old you are as well. Anyway, so that's the extent of the neuroscience that we're going to talk about. So just know that we see alterations in areas of the brain like the prefrontal cortex, amygdala, limbic system. This is tied to the stress response, and we see altered kind of hypothalamic pituitary axis signaling as well. Does anyone have questions about that? Otherwise, I'll move on to the actual pharmacology. Again, just sounds like a lot of information, but just know those basics that I just mentioned then. So what are anxiolytics? Well, these are drugs that, in a nutshell, or broadly speaking, are used to treat anxiety. And there's a variety of different classes of anxiolytics. So I've got them listed here. So a lot of antidepressants. So we talked about selective serotonin reuptake inhibitors, tricyclic antidepressants, monoamine oxidase inhibitors. These also have an anti-anxiety effect. So this goes to show you that comorbidity between depression and anxiety. So they work on some of the same systems and brain regions. There's also benzodiazepines. This is what we're going to talk mostly about today. Also gabapentin and pregabalin. Buspirone, again working on serotonin, has an anxiolytic effect. Antipsychotic agents, so things like quetiapine, can be useful to treat anxiety, can be useful to treat insomnia as well. And then beta adrenoceptor antagonists as well can also have an anxiolytic effect. But mostly you see, again, antidepressants, anticonvulsants, and then benzodiazepines have a very strong anti-anxiety effect. Sorry. So before we talk about benzodiazepines, we're going to touch on the GABAergic system again, because benzodiazepines work on the GABAergic system. So just as a refresher, GABA is the major inhibitory neurotransmitter in the nervous system. It's kind of the opposite to glutamate. So where glutamate, it's released, it causes action potentials and excitation. GABA does the opposite. It's released and it stops firing. It inhibits brain regions. And there's two different kinds of GABA receptors. You've got ionotropic, which are ion channel ligand-gated ion channels. So GABA binds to it, you get chlorine going into the neurons, which hyperpolarizes them. And then you've got metabotropic, which are G protein coupled receptors. We're mostly going to talk about GABA A receptors. So this is a subtype of ionotropic receptors. So what happens is that when GABA binds to the GABA A receptor, this opens chloride ion channels. Remember, chloride has a negative charge. So if more negative ions are moving into the cell, you're going to hyperpolarize it. Okay? You're going to make it more negative, so it's less likely to fire in action potential. And like I was mentioning, benzodiazepines work on these GABA receptors. So benzodiazepines are one of the most commonly prescribed anti-anxiety medication. And what they do is they enhance GABA A receptor function in the central nervous system. It's what's known as a positive allosteric modulator. I don't know if you've heard of those before. But basically what they do is that benzodiazepines don't bind to the same site that GABA does. It binds to another site on the receptor, and that alters the ability for GABA to bind to that receptor. So it enhances the binding of GABA to that receptor. And this is just showing you one of these benzodiazepines that's being applied to cells. So this is what is known as patch clamping. So basically what these people have done is that they've got an electrode, they've basically stuck this electrode in a single neuron, and then you're able to record the current of these neurons and voltage of these neurons. So this is showing you current here. So basically in control, you have GABA puffed onto the neuron, and then this causes a hyperpolarizing current, and then this decays over time. But if you have benzodiazepine, you still get that same, when GABA is released, you still get that same inhibiting current, but see, it takes a little bit longer for it to return back to baseline. So this GABA channel is open just a little bit longer, so you get a little bit more inhibition. There's more inhibiting current going in. So it's affecting that receptor to have an increased inhibitory response. And like it says here, it prolongs the decay of the inhibitory signal, so it stays active for a longer period of time compared to if you don't have it. In some cases, it can also increase the amplitude. So when we're talking about amplitude, we're talking about the amount of current. In this case, it doesn't show you a change in amplitude. It seems to go to the same kind of level. But in some cases, some benzodiazepines can actually increase this deflection. So you might get a current that, ends here and then decays as well. So it increases the amount of either maybe more ion channels are opening or more chlorides going through. So you get a stronger inhibitory current. Again, if you have questions, again, because I know that a lot of you probably haven't come across electrophysiology before, so if you need clarification, just raise your hand. But yeah, benzodiazepines are a large class of drugs, and they all have kind of different properties. So this is just showing you a table here of some common forms of benzodiazepines, so midazolam, zolpidem, lorazepam. As you can see, a lot of these drugs have different half-lives. So they're bioavailable in the body for different periods of time. So this one here lasts, its half-life is 2 to 4 hours, whereas this one's diazepam is longer lasting, so 20 to 40 hours. Some of these just work on the GABA receptor by themselves. Some of these need to be metabolized by the body to have their kind of therapeutic agent. So in this case, midazolam needs to be metabolized by the body, and then that metabolite that's produced actually has that therapeutic effect. So they work on kind of different kind of drug mechanisms as well. And this is just showing you again some of the effects of different benzodiazepines. So some of these can be sedative or hypnotic-like, so they can cause hypnotic effects. Some of these can be used as anticonvulsants, so like anti-epileptic drugs. Some of these can just be straight anti-anxiety. Some can be used as skeletal muscle relaxant, some as anesthesia. So even though we're talking about benzodiazepines in the sense of anti-anxiety, they can, cause a variety of different effects, just like those antidepressants that we were talking about as well. So it's not just relegated to one system or one disorder. It can have multiple effects. So now looking at what does it actually do to the receptors? Well, the GABA A receptor, there's a large heterogeneity in the GABA A receptors. So they're made up of different subunits, and there's many different subunits that make up these receptors. There's 7 different subunit families, so you can have the alpha, beta, gamma, epsilon, omega, all these different kind of subtypes that make up the receptor. So this one here is showing you a beta subtype on the receptor, alpha, all these different ones, and you can have different combinations, and that all affects kind of the channel kinetics of the GABA receptor. Most GABA A receptors are composed of these subunits here, so as you can see in this diagram, and then again, this changes the receptor kinetics. And the expression of these GABA receptor subunits can actually differ depending on the brain region. So again, this is showing you a rat brain, and wherever you see more heat or more red, it means that there's more of that specific receptor subunit in that brain region. So things like an alpha A subunit, it's more expressed in the cortex, prefrontal cortex, a bit more in those subcortical, possibly limbic areas. Alpha 2 expressed a lot in the olfactory bulbs, which is important for the sense of smell, less so in the cortex. Whereas this alpha 5 is, again, mostly in the olfactory bulb and the hippocampus. So it's just showing you that if these drugs are selective for different subunits, you can cause different effects, and you can have different kind of inhibition as well. So this is just, again, showing you how benzodiazepines work. So this is looking at a top-down view of a GABA A receptor. So let's just say you're just looking down at one. You'll see the receptor. It's got its chloride pore, so that's where chloride goes into the neuron to cause that inhibition. And there's all these different receptor subtype units that it's made up of. So we got the alpha, we got the beta, we got the, I think Y is gamma, I can't remember off the top of my head. But what benzodiazepines, why they affect the receptor, again, they're a positive allosteric modulator. There's actually a binding site for benzodiazepines on the alpha Y subunit surface. So benzodiazepines, if there's an alpha Y subunit, it binds to that subunit, and then that affects whether this channel is going to open or close in a specific way. So barbiturates, which we'll talk about later, also have their own binding site, and then GABA has its binding site as well. So this just affects the ability for GABA to bind to the receptor, and then affects the way that chloride goes into the cell. And again, depending on how these subunits are made up within the receptor, a benzodiazepine can cause sedation, it can cause amnesia, it can cause anxiolysis, it can cause muscle relaxation, it can cause anticonvulsant. All very complicated depending on the receptor subunit construction. But don't worry too much about it, just know that this is the binding site for the drug, and that it can affect the chloride. Okay, so benzodiazepines and anxiety. So we established if you have benzodiazepines, they can bind to the GABA A receptor. So then when GABA binds to its receptor, it basically changes the amount of chloride going into the cell, which can affect its inhibition. But benzodiazepines also have kind of a caveat that you actually get a lot of tolerance with benzodiazepines. So the more that you take them, kind of your body, again, develops that tolerance to it. So it seems to be less effective, and you need to kind of up the dose. It's unclear how tolerance develops for benzodiazepines, but it might be due to a loss in this GABA A receptors and or subunit composition. So this is a diagram, and this paper goes into some of the mechanisms why we think tolerance develops. But there's a few different kind of hypotheses of why this happens. So one of these is that the more that you take benzodiazepines, there's evidence that you get removal of the GABA A receptors, or it gets internalized. So there's kind of less GABA receptors present on the postsynaptic membrane. So even though GABA is still being released, there's less targets present at the synapse to cause that inhibition. So you need more of the drug to cause that effect. And this is just showing you it here, so it's being internalized. So that's one mechanism. Another mechanism is that it can phosphorylate different subunits, and if it phosphorylates the subunits over time, then you might get kind of different receptor kinetics. So it might not be as effective or compared to when it first was. Another hypothesis is that it can uncouple the GABA A receptor. So when benzodiazepines bind to these receptors, over time they might affect the ability for GABA to bind to its receptor, so it just interferes with it in some capacity. And sometimes it can actually change the specific subunits that's present on the receptor. So before, where it had, alpha, beta, this Y1, maybe this Y1 will be changed in some way, and maybe it's epsilon, for for example, and then that changes the receptor kinetics. So just different mechanisms there, but that's why we think tolerance develops. There's evidence for all of these with continued benzodiazepine use. And benzodiazepines, they can also have a lot of unwanted side effects if you stop taking them very quickly. So let's just say you stop taking benzodiazepines kind of cold turkey after you've used them for a while. Patients, they can experience very heightened anxiety and a lot of maladaptive side effects, so headaches, tremors, sleep imbalances. So it's really important in people that might have benzodiazepine tolerance or have been on it for a long time that they have a kind of gradual decrease in the concentration of this drug, so just to avoid these unwanted side effects, so kind of gently weaning them off it. And because this is a kind of psychoactive drug, people can become dependent on benzodiazepines. And it's also really dangerous because it can actually, well, it's abused in some sense, in some cases, in combination with opioids or alcohol. It can be used kind of illicitly because of these anxiolytic properties. And it has a really strong interaction with alcohol. So this is showing you here the GABA receptor. So benzodiazepines bind to the GABA receptor to change the amount of inhibition. But alcohol is also a positive allosteric modulator of the GABA receptor as well. So alcohol on its own changes the amount of inhibition through the GABA receptor. So if you have benzodiazepines and alcohol binding to the same receptor, you can get a really, really great increase in the amount of inhibition, which can cause those kind of very high kind of inhibition in the central nervous system, and you get these very kind of dangerous effects where you can get, people getting momentarily paralyzed and unable to breathe, things like that, because they have so much inhibition. But this can be used, people take this recreationally because they enjoy those effects, but it's very, very, very dangerous. So that's benzodiazepines. So there's another positive allosteric modulator of GABA A receptors. This is stressum, or the common name is stressum, but its chemical name is etifoxine. This is similar to benzodiazepine. It binds to the GABA A receptor, but it binds to the beta subunits. But again, it changes the inhibition of the receptor, of the GABA receptor as well. And basically what it does is that it increases the affinity for GABA binding sites, and what it does is it causes a left shift in the EC50 curve. So you've probably heard of the EC50 curve, but it's basically what it's saying is that how much concentration of an agonist do you need to cause a response halfway between baseline and maximal responses. And what happens is that when you take this etifoxine, basically it causes a left shift in that curve. And what it does is that it affects these GABA currents again. So this is again when you've patched on to a neuron, so you've got an electrode within a neuron. What happens is when you puff on GABA, so that inhibitory neurotransmitter, if you look at the red, in a normal sense, you get this very quick hyperpolarizing current that decays over time. And when you give more of the GABA receptor or of the GABA neurotransmitter, you get an increase in the amount of inhibition, but it resolves very quickly. But if you have etifoxine present on the receptor at the same time, as you can see here, with just 10 micrometers of that GABA micromolar of that GABA, so a low concentration, you get a very high increase in that inhibition. So with that same amount of GABA that's being puffed, look, you get a very high or increased amplitude of that GABA response. So you're getting more inhibition, and it decays over a longer period of time. So again, more inhibition, and you're getting effectively that. See this red here, this is when you puff on more of that GABA neurotransmitter, so 200 micromolar. You're getting that 200 micromolar effect just at 10 micromolar. So it really affects that receptor to cause more inhibition, which is explained there. So it causes that left shift in the curve, you get more GABA inhibition with less neurotransmitter present, so it really affects that amplitude. It's also got some other effects. It's been shown to increase the entry of cholesterol into mitochondria, and it also is converted into kind of another metabolite that is also an anxiolytic enzyme as well, so it seems to affect multiple mechanisms. Anyway, that's the two kind of anti-anxiety drugs that we'll talk about. So again, so etifoxine and benzodiazepine. Just know how benzodiazepine works compared to etifoxine and why they might be used to create an anti-anxiety effect. So moving on, there's also another class of drugs that can be used as anti-anxiety as well, but they're kind of their own class, which are hypnotics. And these are drugs that treat insomnia. So they induce drowsiness and promote sleep onset and maintenance. So they make you fall asleep, essentially. But they can also have an anti-anxiety effect and even an antidepressant effect in some cases. So these are different from sedatives. So you might think hypnotics and sedatives are the same thing. They're kind of a little bit different. Again, hypnotics induces drowsiness and promotes sleep, whereas sedatives kind of reduce activity and alertness and kind of induce a state of calmness, whereas hypnotics kind of make you more sleepy. So there's a distinct difference between them. And hypnotics are usually used, again, to treat insomnia. So insomnia is the inability to sleep. So a lot of people that experience other mood disorders or neurological disorders typically have insomnia as well. So they're unable to sleep. And sleep, again, I don't need to stress how important sleep is, not just for memory consolidation, but just your general alertness and cognitive function. And again, there's multiple kind of systems involved with insomnia. We know some mechanisms of what induces sleep, but again, a lot of these different monoamines and different brain regions all have a hand in it as well. So if you have some alteration in some of these brain regions, it can really affect sleep in a very complex way. In hypnotics, just like there's a lot of classes of benzodiazepines, there's a lot of kind of different drugs and classes of hypnotics as well. So benzodiazepines can have a hypnotic effect, but they're more kind of like a short-acting effect. Some other class of hypnotics are Z drugs. Again, these are short-acting. They work on that GABA-A receptors as well. They have little anxiolytic effects, but they have more of a stronger hypnotic effect. There's melatonin receptor agonists, and we'll talk about this very shortly. These are shown to be effective in treating insomnia, especially in the elderly, children that have autism, and also individuals with blindness as well, because some of the cells in your eye are very important for controlling your sleep-wake cycle. Hypnotics, there can also be other positive allosteric modulators of that GABA-A receptor. Again, they bind to different sites from the benzodiazepines, so just contrasting with benzodiazepines which bind to that site. There's also orexin receptor antagonists. I don't know if you've heard of orexin before. If you're doing neuroscience, you hear orexin pop up every now and then. Orexin is very important for regulating your sleep-wake cycle. So if you've heard of circadian rhythms before, it's your 24-hour body clock. Orexin is really important for kind of controlling this 24-hour body clock. When you get tired, the sleep pressure build up, all those kinds of things. We won't talk about it because it's very neuroscience heavy. But also antihistamines can have a hypnotic effect as well. They work on the histamine receptors, so they induce drowsiness and sleepiness, but they also show potential as being a straight-up hypnotic drug as well. So while it might not be used for the hypnotic effects, it's more to block those histamine receptors, it can also induce drowsiness. So there is some evidence to suggest it might be, there's promise on its own as a hypnotic drug. So one hypnotic that we will look into a bit more detail is barbiturates. Barbiturates can also be used as anti-anxiety drugs as well. Similar to anti-anxiety drugs, they depress the nervous system, but they're a little bit more dangerous, I suppose. They have a smaller safety window, and this is because barbiturates have a very strong effect on the respiratory system as well. So some people use it as an anesthetic in some cases. In animal studies, people use it as an actual euthanizing agent. So veterinary clinics use it to euthanize animals because it has such a strong effect on the respiratory system. But if you use it in small doses, it can be used as a hypnotic, but just know that there's a kind of smaller window for when that can get into the kind of dangerous territory. And there's, again, different types of barbiturates, just like benzodiazepines and anxiolytic drugs. Some of them are very, very short acting, so they cause a hypnotic effect within 5 to 15 minutes. Some have a longer effect, like pentobarbital, 3 to 8 hours. This is usually, like I was saying, the euthanizing drug. And some are also long acting, so they can last days. So depending on the drugs and kind of their characteristics, you can induce a hypnotic effect for a short or a long period of time. So how do these barbiturates work? Well, just like benzodiazepines, they bind to an allosteric site on the GABA A receptor, but it's a site that's different from the benzodiazepine site, and its action is a little bit different to benzodiazepines. So let's just unpack this first. So this, again, is looking at that top-down view of a GABA A receptor. This is that chloride channel. These are all the different subunits. So as you can see here, GABA binds to that specific site that I showed you before. Benzodiazepines bind to its own specific site that changes this channel in a certain way. And barbiturates also have their own distinct binding site as well that affects that GABA A receptor. Now, how are barbiturates different from benzodiazepines? Okay, so this here is looking at, again, this is where we've got an electrode in the cell and we're recording its electrical activity. Before we were looking at the current, so the ions moving in or out of the cell, which generates that inhibitory or excitatory current. This here is looking at the voltage. So we're not looking at the current moving in, we're just looking at the voltage of the cell. So is it a bit more positive or a bit more negative? We're just looking at the voltage changes. In a control cell, so you're just recording the activity of a cell, the voltage is pretty stable, okay? Stays pretty stable, tiny little fluctuations. If you puff on GABA, so the inhibitory neurotransmitter, you see these kind of changes in the voltage. It gets a little bit more negative. So it's going negative, going back to baseline, but basically what it's showing you is that it's just getting a little bit more negative, okay? Because those GABA neurotransmitters are opening those chloride channels, chloride's moving in, so you're making the cell a little bit negative. That's what it's showing you here. Now, if we have benzodiazepines, again, benzodiazepines, like I was saying, affect the inhibition, and you see basically it's a little bit more negative for longer periods of time, okay? So what's it actually doing? Benzodiazepines are affecting the opening of this chloride channel, okay? So basically what you're doing with benzodiazepines is you're just making that channel open just a bit more often. You're not increasing the amount of time it's open, you're just making it open and close just a little bit more, okay? So where GABA might, open it like open, 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 benzodiazepines will make it open, 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 open. It just basically increases the frequency that chloride channel is open. So you're getting a lot more negative voltage in the cell. So it's a bit more inhibited because the chloride channel opens more frequently. Now, when you have a barbiturate binding to the receptor, you see similar things. It's getting more negative, but see how this stays negative for a longer period of time? So here it's going, opening and closing, it's going negative baseline, negative baseline. This is going faster, negative baseline, greater frequency. This, once it's open, it's negative for a longer period of time. It's not fluctuating. So they work on two different mechanisms. Benzodiazepines increase the frequency of the opening. This one, once it's open, it keeps it open for a longer period of time. Okay, so two distinct mechanisms. Does that make sense? Because this is really important, just knowing the differences in, and if you can't read these, feel free to ask questions, but again, hopefully that explanation suffices, but again, if you have questions, please ask me. Now, barbiturates, they also, at higher doses, they can also have a distinct effect on GABA receptors, and they can increase chloride conductance as well. Don't worry too much about that, but just worry about these kind of two mechanisms more so. And that's just explaining what I was saying there. So they increase the amount of time that the channel's open. So there's also evidence to suggest that when you have a high concentration of barbiturates, it can also act on voltage-gated chloride channels. So not just like those GABA A channels, they can just affect voltage-gated chloride channels in general. And then they can also inhibit the release of glutamate on those neurons as well. So they can have an additional effect and just affect those chloride channels in general at high concentrations. So they can have a really strong inhibitory effect. And they're also been shown barbiturates to inhibit those AMPA receptors. So the AMPA is the excitatory receptors. So they can actually stop those excitatory receptors from opening as well in some cases. So they can have a bit of a stronger and kind of less specific effect. So not just affecting that GABA receptor, they can affect those excitatory receptors as well in some cases. So that's why we're saying it has a kind of, you need to be careful with the safety profile of it, because if you have too high concentrations, you can affect multiple systems, which might stop your breathing, things like that. All right, and then to finish up, we'll talk about one other class of hypnotics, which are melatonin receptor agonists. So I'm sure everyone here has probably heard of melatonin in some capacity. Usually see it in the pharmacy, just over-the-counter drugs, things like that. We usually say it improves sleep. So how does melatonin actually work? You might be, I don't know if you're interested, but melatonin is really important in the sleep-wake cycle. So again, we have our own internal body clock, and over the course of a day, the amount of melatonin increases, which increases our sleepiness. Usually it coincides with darkness. So when the sun starts setting, the lights start turning off, you see a general increase in the amount of melatonin released, and that causes that sleepy effect. Now, melatonin is released from the pineal gland. So this is just showing you side view of the brain that's been cut. In the brain, you have ventricles, which are kind of these empty spaces that have fluid going through them, which helps with insulating the brain, shock absorbance, and also moving waste around the brain. Excuse me. And the pineal gland is located just near one of the middle ventricles, the third ventricle, which is just this little structure here. And this releases the melatonin. Now, the release of melatonin is controlled by the suprachiasmatic nucleus, which is located next to the hypothalamus. And the this subchiasmatic nucleus basically controls the release of melatonin from the pineal gland through your eyes. So you have neurons in your eye that help see everything and turn that light signal into electrical signals for us to understand it. But there's a specific population of cells in the eye called melanopsin that have a melanopsin protein. And these basically are a specific population of cells in your eye, and they project to that subchiasmatic nucleus. And basically what happens is that subchiasmatic nucleus is then getting information on the light levels in the environment. So when there's less light, your suprachiasmatic nucleus is like, okay, we're getting into darkness. Let's tell this gland to start producing more melatonin so we can go to sleep. So basically eye to the nucleus tells the pineal gland to increase melatonin, broadly speaking. And this is just showing you in a person the amount of melatonin that's released over the course of a day. So this black line is looking at melatonin in plasma, I think it is. So just looking at if you take some blood from someone and you just look at the amount of melatonin that they have. Most of the melatonin peak starts increasing around evening, early nighttime. So about 7, 9 o'clock, you start seeing increases in melatonin, and this melatonin peaks around about 2 a.m. So again, it's just inducing that sleepiness, and then it kind of tapers off over time, and then you start waking up. This blue is showing you melatonin levels in saliva, so you can also detect it in the saliva, not just the blood, and it's showing you the same thing. You get this increase around evening time. And then this red line is showing you this thing here. So this is a metabolite of melatonin, and this is looking at the metabolite in urine. So you can also measure it in urine. So it's basically showing that there's more of this metabolite in urine because there's more breakdown of melatonin because it's circulating in your body. So just showing you where melatonin peaks. Now, melatonin has its own receptor. So melatonin receptors, which are G-protein coupled receptors. So you have M1 and M2 receptors. These are mainly linked to the GI pathway. So when melatonin binds to these receptors, it typically causes an inhibitory effect. And this inhibitory effect is really important in the certain brain regions that it projects to cause sleep. And this is just showing you a cartoon here. So you've got the melatonin receptor 1, melatonin receptor 2 subtype. Most of them work to reduce cyclic AMP. which is that inhibitory response. Binding of this receptor also changes voltage-gated ion channels as well. So the potassium channel, basically it opens potassium channels, so more positive potassium is moving outside the cell. So remember, if positive ions are moving out of the cell, that means it makes the cell a little bit more negative, so it inhibits it because there's less positive ions in the cell. And it can also change the amount of calcium going into the cell as well. Yeah, so these receptors, again, mostly inhibitory. Melatonin binds to them, causes inhibition. And this also feeds back to genes that are really important for affecting our sleep-wake cycle as well, but we won't go into that. And these melatonin receptor agonists are useful because, again, there's very low abuse potential. So, again, you can get melatonin over the counter kind of thing because there's not really any strong side effects like barbiturates or things like that. So there's very low abuse potential. There's no rebound insomnia. So if you take melatonin, some people feel a little bit more groggy the next day, but generally you don't have a very strong sleep deficit or need to fall asleep a few days after you've taken it. Little residual effects, and there's no evidence that there's actually dependence or withdrawal symptoms as well. So that's why it can be very, very useful, and you probably hear it a lot. Melatonin's really great to use. And this is just out of curiosity that you might be interested in as well. But melatonin is also produced from serotonin as well. So again, that monoamine system as well is kind of implicated with melatonin and melatonin receptors as well. And in this diagram, it's showing you that tryptophan, which we talked about before, is converted into serotonin, which gets converted into melatonin. So you might have heard people say things like, if you increase the amount of tryptophan, because we can get tryptophan in different foods like bananas, things like that. So people say, if you eat bananas just before bed, you might induce the production of melatonin, so it might make you fall asleep a bit more. And I remember one of the old unit coordinators of one of the pharmacology units used to always say, eat a banana 2 hours before bed because it'll make you fall asleep. Don't know if that's true. There's not much evidence that I've seen that suggests that increasing dietary tryptophan increases serotonin or melatonin, but it's just useful to think about as well. Anyway, that's it. So thank you for coming to the lecture. Again, anxiolytics and hypnotics. Next Monday, we're going to follow up these lectures with looking at Alzheimer's and dementia and kind of the therapeutic drugs that we can use in those neurological diseases and disorders. And then after Monday, Alex Tang will finish off the lecture series, and that'll be it for neuropharmacology. So thank you very much. Hope you have a great weekend, and I'll see you on Monday.